All right, it is recording, so I will share my screen. All right, so hopefully now you should all see the course schedule on your screen. Give me a thumbs up for those of you who got a video running. Perfect, thank you very much, I appreciate that. So today is the 15th, we've got our lecture. After lecture, your SmartBook number 3 is due. Hopefully you've already read that so that you can participate in class today, because we're going to be covering all of chapter 3 today during lecture. And you'll also see that come this weekend, starting Friday, is when your first exam opens. So these exams, students often get a little worried with the first exam. They're not sure what to expect, that kind of thing. So I have actually provided you a study guide. There are actually study guides for all our exams. But what that does is, well, maybe I'll open the study guide too. That would probably be helpful as I'm talking about it, huh? So let's just make sure I don't open up the exam itself. There it is, right over there. I could open that, then you'd see some of the answers. So here's the Unit Exam 1 study guide. For all of the study guides, there's a couple open-ended questions and then some multiple choice questions for the study guides. The entirety of the exams are multiple choice. So you'll see either these exact questions on the exam or ones very similar to it. So like this one, in an experimental study, the effects of different levels of anxiety on self-esteem. Anxiety would be the what variable. So you just answer in those multiple choice. The entire exam will be done through Canvas. It's essentially just programmed as a quiz on Canvas. So on Friday, well, I guess I should mention these open-ended questions. So if you see these open-ended questions, review neuron structure and function. Essentially, I'm saying, hey, there's a couple questions there on the exam about neuron structure and function. There's a couple questions about plasticity. Also, remember, feel free to raise your hand at any time during the class, and then I will be able to answer any questions. So there will be multiple questions on the IRB, multiple questions about epigenetics, which we're actually going to talk about in a few minutes today. So these questions, you can expect to see multiple choice, a few multiple choice questions on each of them. And all of the multiple choice questions on the exam are in a similar format to these. So 7, 8, 9, 10, 11, 12, 13, 14. You'll see a lot of questions like that. If I remember right, exam 1 is about 60 multiple choice questions, and you have, I think, an hour and a half, but I'll have to double check. And then I'll send an email reminder before Friday so that you know the exam's coming, the time limit. So the exam opens at essentially right after midnight on that Friday. So you can take it anytime Friday, you can take it anytime Saturday. Just make sure you open it up and start taking it in enough time that you can finish it before 11:59 p.m. on Saturday. So if you open it up at 11, make sure that you can finish it within that hour. If you're opening it up at 11:30, you might not have time to finish the exam, and you might end up getting kicked out, and only the answers that you've completed will be counted. So you'll have that time limit during that. I'll actually go and check what time I've allotted. I think I'm completely prepared, and then I'm like, oh yeah, I should have double checked that before the class too. 70 minutes. So there are, why is it not telling me how many questions here? So you'll have 70 minutes for the exam. It's 100 total points, so some of the questions are worth, almost all the questions are worth almost 2 points each. But there's about 60 questions. All right, so again, that exam will open Friday, so you can take it anytime Friday, you can take it anytime Saturday. If you know you're going to be out of town on, say, Friday or Saturday, that you're going to a wedding or something and you're not going to be able to do the exam, let me know in advance, so then we can open it up, say, tomorrow or Thursday and get you prepared beforehand to take that exam. Another thing that you'll see is your question paper will be due on Friday as well. So that's basically right before midnight, your first question paper is due. And I did send that link with that video, so a lot of what I described about question papers will be repetitive right now. but I wanted to talk about question papers too. But before I do that, are there any other questions about the format of the exam? Essentially what you'll see, another thing that I think could be helpful is right now my Canvas looks a little different than yours. You probably don't see any block for the unit exam, but on Friday, this block will open up. It'll show unit exam 1 is available. It will give you a due date. You'll just come down here and click it, and you'll do the exam. So I can preview it actually. And this is more similar to what you will see. So you can skip through the questions. You don't have to go in order. Oh, there we go. 54 questions. So about 60. Like I said, you'll have 70 minutes. You'll go through, choose the questions after you've completed the exam. It should, as long as it's programmed correctly. I'll double check before the exam, but it should show you which answers were correct and which were not. It gives you your time that you've got left. I still have yet to receive any disability services changes for any students. So if that's something, let's say in previous classes for say something like ADHD or anything like that, you've had extended time for exams, definitely get the Students Disability Center to send me an email and let me know how to modify exams and other assignments for you. I haven't received any of those yet, so if I should have from you, definitely contact them, contact me as well. So any other questions about the exam, the format, anything like that? It's just a whole bunch of multiple choice questions. Oh, and I'm still showing the exam questions. There you go. Well, you can prep about 12 questions. You've seen them now. So my wife just heard me do that in the background. She's like, oh no, it's fine. 12 questions, that's not the whole exam, right? And I mean, hopefully you already are studying for them anyway. I want you to do well on the exams. I don't want students to fail. I try to have a good number of pretty easy questions, moderately difficult, and then a couple really difficult questions. Looks like we've got one question from Dawn. Go ahead.
Yes, I was just wondering if you had any options for a retake or if that was it, like the test you took was your final grade. I did read the syllabus, but I read so many and so I forgot. I apologize.
No, that's all right. And really good question. Yeah, and you can also check the syllabus for this as well. There's not any retakes in the class, so definitely prepare before the exam to do as well as you can on the exam. If there's something strange, like say your internet cuts out and you're not able to get back in and do the exam, then I'll let you redo the test. But let's say you get a poor grade, like you studied, you thought you were prepared, and you get, say, a 40%. In that kind of case, I don't have retakes for the test. But if it's something like my internet totally died in the middle of the test, I couldn't actually finish it, something like that, I'll let you retake for. So yeah, really great question.
Thank you.
Yeah. Any other questions then? So it looks like, Don, your hand is still up. So if you still have questions, go for it. If not, feel free to put your hand down whenever you're done talking. I just don't want to put it down myself and ignore you if you did still have questions. All right, any other questions about the exam before I talk a little bit about question papers? All right, so the exams are a fairly large portion of your grade. There are 100 total points towards your final total grade. So definitely study well for them. One thing you could do is even meet with other students in the class remotely and do some kind of study group. So if there's students who today during the class, you're like, oh, that student seems to know what they're talking about, or they answered this question well, maybe email them and see if they want to join a study group or something like that. We're not going to go over the study guide before the exam. So that could be something that you do together as students, like outside of class. If you have, say, 1 or 2 questions on the study guide, so I'm not going to send emails to every student answering the entire study guide, because that's part of the purpose, is to study and find the answers yourself. But if there's 1 or 2 questions that you're like, I'm really not sure if it's C or D on number 12, feel free to ask me 1 or 2 of those kinds of questions. All right, so let's talk about question papers. Oh, I've got emails up. That's not what I was meaning to do. Here we go. So one thing that you should do for question papers is read over this entire document. I'm not going to read this to you on screen. That would be very boring for you and me. So I'm going to basically summarize these things, but this gives you a lot more detailed instructions on what to expect from a question paper. We will go in a little more detail over an example question paper. So essentially what a question paper is, well, I will show you an example of a question paper. First, I think that could be helpful. So this is an example of a question paper. You should see that on your screen. It is a question that you have that maybe has come up in your reading, something that's interested you. Maybe you're reading about neurology or reading something about sensation and perception for today and had some more questions about it, wanted to know more about visual perception, wanted to know more about color blindness, wanted to know more about how neurons actually connect up to other neurons, things like that. So anything that from these first 3 chapters that have interested you, it's essentially a paper that you write, a 1-page paper about a question of your own interest. So as you can see, this one is actually 2 pages, but yours is only required to be 1 plus a page of references. So here, my question paper example here is, what are the possible negative effects of Ritalin? So maybe in reading about certain drugs to treat different disorders, it might have been something that interested me. So what might some negative or even positive effects of Ritalin be? So here I start out, I have some information about Ritalin. As you can see, I have some citations in here. So these are called in-text citations because they're inside the text. So this is essentially saying Greenberg had the original source that told me facts that I put in this line. So Ritalin is a fast-acting drug, lasts between 2 and 5 hours, depending on the person and dose, which means parents of children and adults using the drug need to carefully monitor when they last took a dose to ensure symptoms of ADHD don't emerge again. So a lot of that information came from this source, Greenberg. And so that's one part of these question papers, is finding sources to answer your questions. And usually these are going to be peer-reviewed journals, but we can also use websites. So I have a question, I look for sources. So I guess that's kind of the timeline of a question paper. What is your question in psychology, in the topics that we've covered? Even if it's kind of tangentially related would be fine. So you wanted to know more about one of the people it mentioned in the chapter on neurology. So maybe you wanted to know about Phineas Gage and wanted to look up some more information about him. That could be a good source for a question paper. If you're having trouble thinking up topics for a question paper, I can give you some pointers, but hopefully the whole discussion today gives you some good ideas. So I've got a question. What are maybe some possible negative effects of Ritalin? So the next thing I would do is look for sources. And you can see with my reference list here, I have quite a few sources. So some of these, that Greenberg that I just mentioned is actually a website, the APA website. It's an article on there about, well, this is the title, what's the treatment of child and adolescent ADHD? So basically talks about that. So I found this on the APA's website, so I can cite it as one of my sources for helping to answer my question. So that's the first thing. Find a question that's of interest to you. Go find some good sources. So the APA website is a good place for that. Another good place for that is journals. So you see International Journal of Drug Policy. You probably don't know where to find that, and that's okay. I'm going to show you in a minute. Another source from the APA website. So APA is American Psychology Association. I don't think I said that. I've got some sources from WebMD, some from the National Institute of Health. So depending on the question you're asking, you could get sources from psychology, medicine, sociology, history, philosophy. There's a lot of different other fields that, like we've talked about, tie into psychology that you could find some good sources of. So the requirement for the paper, though, is again 1 page long in answering your question and at least 2 sources. So here I've got, what is this, 5? 1, 2, 3, 4, 5, 6. So I can count. So 6 sources, but you're only required 2. One of those has to be a peer-reviewed journal article. So something like this, the International Journal of Drug Policy, that's a peer-reviewed journal. Another source can be a website. So I think this actually goes to just the APA Monitor rather than the actual article. So that was something that I should have changed and gone to the exact article itself. But the APA is a good place to find some good information to use for these question papers. Now let's also look at the library. So to get to the Salt Lake Community College Library, it's really quite easy. Salt Lake Community You can search for it. You can link to it through the colleges. So here we go. So this is where you can find peer-reviewed journal articles. So here's our library. It looks a little bit different than the last time I looked, but it's really easy to find the research databases. So you're here on the main page. You come down here to collections, and this first link is research databases. So you could click there. You could also click journals, but research databases is what I recommend. And there you can go and select through a bunch of different journals. So for most of the questions that you're going to be asking, they'll be around the field of psychology. So let's show you 2 of probably the best. One is going to be PsycInfo and the other is going to be Google Scholar. So I'll go to P. Oh, it is there. So you can scroll down and find PsycInfo. So what PsycInfo is it is an EBSCO database. I don't remember what EBSCO stands for. Basically, PsycInfo is almost all psychology journals from 1887 to current. So if there's something that's come out in the most recent month or 2, you might not find it, but if it's been out like 6 months or so, it should be in PsycInfo. So that is where you would want to go to search for anything psychology related. You'll have to log in. If you're on campus, it'll probably automatically log you in because you're already connected to the networks. But if not, you'll have to log in. And then you'll get a page like this. So here, this is just a database that you can search. So I might look for Ritalin. Maybe I'm interested in, oh look, Ritalin side effects. I can select that. And I can search for journal articles this way. PsycInfo will also, let's do that. You don't have to change that. This has been added recently. So I'm going to say, come on, take me to the information. Sometimes it makes you add things to your search. And okay, so I've now added a couple things to my search, and it has come up with 13 results. I'm actually going to delete this pharmacology. Let's try that again. Ritalin in children. Let's see if I can search just for Ritalin in children. All right, here we go. Sometimes it does that. It didn't used to. Sorry about that. But here we go. Efficacy of Ritalin in ADHD children under neurofeedback training. You can see here, this is an academic journal. So that would probably work. Most likely that is peer reviewed. What you want to make sure is that you have this link to full text articles. There's not a required publication date because your topics could be so varied. If you're looking at info on Freud, you might actually want to read a paper that Freud wrote, and so that would be a little older, right? So you don't have a time span, but for something like Ritalin, you can probably move the publication date up. This one, you can probably get newer things. So for Ritalin, you could probably find a lot of good stuff from 2005 on. So that first article is right there. And so what you want to find is something with full text so that you can open the entire article, and you can also make sure that you're looking only at academic journals. And it'll update. Now you'll see the search results are fewer. So we might say, okay, I think this article sounds like it might be useful for answering my question. So I might open that one. I might look at this one, and I might look at this one. So I have this question about Ritalin. I open a couple article abstracts, assuming they work. Wants to open them up all one at a time. Well, that's not as helpful for showing the class how to do this, but that's okay. All right, so here I've got the authors. I've got the source, which you'll need all of that to make your citation. The main thing you want to look at is the abstract. So you say, I think by that title, this article might be useful. So let's go and read this abstract. After you read the abstract, oh, I see a good question about websites to avoid. Yes, we can talk about that in a minute. I'll first show you how to go through these sources. But if I forget, Lindsay, definitely remind me. So if this abstract now sounds like it's something that would be useful to you and would help answer your question, give you more knowledge about Ritalin, know about its side effects, then I would say get the full text of the article. So PDF or HTML, either way, is great. So the PDF is just a portable document of the article. So as you can see, the article will have its abstract. It'll have its title up here, the author's over here. Sometimes the abstract might be like small and in the middle, or it might be as part of these columns as well. And then as you read these journal articles, I don't expect you to understand everything in these journal articles. This is just a first taste of what real scientific research, how it's published. So what I want you to focus on as you answer your questions is the abstracts of these to help answer your questions, the introductions, and then if you scroll way down, you should be able to find something that says discussion or general discussion. So if there's multiple studies in a paper, they might have multiple discussions. If that's the case, look for a general discussion. This one has a discussion and conclusion. So if I was to recommend any student going over this article, I would say read the abstract, read the whole introduction. When it gets to something like methods or design or study one, skip that part and skip all the way down to where it starts talking about discussion. Because discussion is essentially where in a little more lay terminology, they kind of describe what results they found. So the results section is going to give you a lot of statistics, what tests they ran. And if you understand that stuff, great, but I don't expect you to at all at your stage in this program. So reading the abstract, intro, and discussion would be great. Just skimming those too can often answer your question. If all you want to get is side effects of Ritalin, you might find all those side effects within a little bit of the introduction. And so that might have answered your question enough that you can write about it in your paper and you don't have to read the discussion as well. So you can skim through these articles. If it doesn't seem like this article will work, go and find another one. So again, for your question papers, you need two sources. At least one of them has to be a peer-reviewed journal article, although both of them can be. All right, another database that I would recommend, especially if you're doing things outside of psychology, is Google Scholar. So we're back here on the library website. So Google Scholar should, oh no, did they take off Google Scholar? Well, that's okay. Even if they did, One thing I like about Google Scholar is that it's free to access for everybody, and you can search for anything and read almost all abstracts and find a lot of full text articles. So let's see Ritalin side effects. Maybe Ritalin side effects in adults. So one thing about Google Scholar is it's pulling from medical journals, it's pulling from history, it's pulling from sociology, psychology, pharmacology. So you're going to find a lot more results. So you're going to need to do more work yourself to try and narrow down and find exactly what you want. So what you'll see is you'll find a lot fewer full text articles, but here is one with a full text. So you might open that one and say, well, this one has full text, maybe this will be useful. So let's say these ones don't have full text, this one does. So this one's from National Institute of Health. And it looks like it's actually a journal article that's just published on the National Institute of Health website. So here's their abstract here, and then they jump into their intro. They don't actually have it like delineated saying abstract. Just know the first thing is going to be the abstract. So this is a policy paper, which could work as a source, could help you answer your question, but this specifically would not work for a journal article. Whereas this was published in a journal, this one would work. Now, how do you make your reference list? Well, Google Scholar actually makes it really, really easy. So let's say this first one was the correct source, was one of your sources you wanted to use. You can go to this little quotation marks, click it, and then copy and paste this APA citation. So that's as difficult as it is to make your reference list, is asking Google Scholar to give you the citation. Then you just paste it in proper APA format. If you're using PsycInfo, you can also get the citation this way. Let's actually go back to the previous page. All right, so here's where we would read the abstract. You can click this little cite button on the right side, and then just make sure you're pulling out the APA citation. So copy, paste that into your paper. As a reminder, your example question paper, the reference list should look a lot like this. So there is a rubric online that goes into more detail. This how to write a question paper, it goes into more detail as well. It gives you a little bit of input here on how to write APA citations, how to put in-text citations, how to do references in here. So generally, I suspect you'll be using websites as well as journal articles. So that would be, so this is how to do a website in APA format as a reference. This is how to do a journal article. Again, journal articles can be really easy on Google Scholar. You can just copy and paste them. The citation itself. As you use a journal article and find some information, if you want to do a full quote, make sure that you put that in quotation marks within your paper. If it's paraphrasing the research, like this line says, in some cases, ADHD can also be treated with antidepressants. So multiple sources confirmed that. So I put both those sources here, that some cases of ADHD can be treated with antidepressants. So this is not a direct quote, but that information came from those sources. So I'm saying, hey, this is where I found that information. And then down here, you can find both Gottlieb and Greenberg in their full form. So you could then go to that if you were reading this article as the professor or someone else reading your paper. So again, question paper is a one-page paper, research paper essentially, about a topic of interest to you that's connected to the topics we've covered in class. Hopefully there's something that's interested you so far in the class, and hopefully you can find some good information on it. Good websites are things like the APA website, WebMD, things written by the researchers. Poor choices in websites would be things like some random person's blog who doesn't seem to have any expertise in an area. News articles for this purpose probably aren't as useful. You might be able to find like one or two facts about something from a news article, but that wouldn't generally be as good. Stick to things where it's written by the researchers themselves or someone's reporting on the research of researchers. Sometimes a lab, for example, that's doing research will have someone kind of summarizing the research that their lab does. So lab websites, journal articles, things like the American Psychology Association, things like that would be really good websites. A random blog by someone who doesn't really have any expertise in an area would not be as good. That being said, there are good blogs. So one example is Darcia Navarez is a developmental researcher who writes a blog about the research she and her lab does. So she publishes a paper on a subject for scientists. Then on the blog, she'll write a couple blog posts about it and really put it in language that, say, a high schooler could understand. So something like that can be useful, but those are a lot more rare. So if you have questions about a source, feel free to email it to me. Be like, hey, is this a good source? Maybe can you point me in a direction of a better source? That would be great. I think I have hit on all the points I wanted to on question papers. If you have additional questions, use the raise hand feature right now. We can talk about it. You might want to also watch that video I linked before and I might have gone over things a little bit differently and made more sense in that video. Yeah, any questions about question papers? Oh, I didn't show you where to upload them. That could be important. Don, go ahead. I see you've got your hand up.
I noticed that there was 2 extra credit question papers. Can we just turn those in at any point, or?
Yeah, that's a good question. You can turn those in. I would recommend waiting till after you've got your grade on your first one so that you can make sure you're doing the question papers correctly. But yeah, at any time in the course, you can turn in those 2 extra credit question papers. I will show you where those are. Again, your Canvas might look slightly different than mine because I see everything and some things are hidden to you. So question paper extra credit 1 and 2, their final due date is November 29th. So that is near the end of this class. So at any time in between now and then, you can do a second or third, some extra credit question papers and turn those in. So again, I would recommend you wait till you see your grade on your first one and then make changes so you can get all of the points. But yeah, those are worth 20 total points each. So that's a possibility of 20 points here and 20 points here, or 40 total extra credit points. And that is one of the only ways to get extra credit in the class is by doing those question papers. And so those can be over any topic in psychology that's of interest to you. So yeah, really good question. So to turn in question paper number 1, you'll go here in unit 1, and then question paper 1 is here. Again, it's going to look slightly different to you. When you click here, you will see a drop box, basically a place to upload a file. Upload your question paper in either PDF, .docx, or .odt format to make sure that I can read it. Sometimes Pages doesn't like to open on anything like Pages files, doesn't like to open on anything except for Macs. So upload it in doc, PDF, or .odt format. And then you will, as you do that, you will see a report that shows how similar your paper is to other papers out there in the internet. Basically, what I want for that is that you can check and see if you have plagiarized. So if you see 14% similar, you click on it and you see, oh, the title, or it's highlighting my citations, you'll know you've got nothing to worry about. If you see it says, say, 60% similar, and you open up your paper and almost your entire paper is highlighted as being the same as a website that you used, that shows that you just copied and pasted, or you didn't paraphrase and you're cutting and pasting too much stuff. If there's a few direct quotes that you have quoted and cited appropriately in the paper, you're good to go. But if a lot of your paper is that way, do make some changes. If you see that, you can resubmit until the due date. So once the due time passes, you can't resubmit at that point. So yeah, I think for real now, I think that's everything about question papers. Again, it should look something like this. You've got about 12-point font, Times New Roman. You're talking about probably the definitions of what you're looking into, Ritalin, ADHD. You're starting to answer your question with sources like from WebMD or a journal article. You can include relevant personal information. So the thing that inspired this question for me was that my mother, as a child, didn't want me to use Ritalin for treatment of ADHD because of things that she had heard about it. That things like my height and weight would be, my growth would be stunted as an early adolescent. And so that's what inspired this question. So you can put that kind of personal information in these papers if you want. You don't have to, but just as an example, you can definitely tie this in. So talking about neurology, you might talk about how you had nerve damage and your nerves took some physical therapy to regrow, and that inspired your question. Something like that would be perfectly appropriate for this kind of paper. Answering the questions should mostly come from the research articles. All right. So any other questions about the question papers before we move on to Chapter 3 material? All right, well, I do think that was actually a little more long-winded than the video I made, but it does give students a chance to ask questions as we go through, so hopefully that was helpful. Let's start off, before we get into Chapter 3, let's actually finish up our content on the neurology and as well as genetics, and how our genes might affect things like neurology and our behavior. So this is from the end of Chapter 2. We'll be talking about heredity environment correlations. So let's jump to that material. All right, as a reminder, definitely know the basic cell structure of a neuron, that you've got the cell body, one axon sending messages. There can be lots of axon terminals, or axon branches, and then there's multiple dendrites on any given axon, and how they connect to each other. A little bit generally about what the process is of how that action potential travels down the length of an axon. All right, so let's talk about genetics and behavior and how it's related to neurology, as well as how it's related to general human behavior as well. So what is a chromosome? You've probably heard that term. If you've had some more advanced biology classes, you probably know this. A chromosome is the thread-like structure in every human cell made up of DNA. So what is DNA? So let's break it down even further. DNA is simply a molecule. I mean, it's a very complex molecule, but it is just a molecule. So it's a structure, the way the molecule is organized, which different pieces of the molecule are following it in a chain, that basically gives us our genetic info. And so what is a gene? A gene is a piece of that DNA, a unit of hereditary information. So basically a piece of that strand of the molecule that tells other parts of your body, proteins, for example, what to do to create things like the colors of your eyes, where hair should grow, where your bones should be, things like that. So what are proteins? They're essentially the building blocks of cells, and they regulate and direct body processes. So a little image might help this make more sense. So you've got the chromosome here, and we've got just a snippet of it zoomed in. So we see this DNA. You can see it's this molecule. And here we've got the adenine, cytosine, thymine, guanine. So that's what the ATCG or ACTG is about. It's referencing those different chemicals. So a protein will then come up and essentially read this DNA, this gene at this portion, see what it is, and then go and tell the cell what to do. So, hey, grow some cells here, stem cell, grow a stem cell. This stem cell will then become part of your skull, or the stem cell will become something else, part of your brain, a neuron. And then overall, these proteins then direct cells and direct our bodies of what to do. So Yeah. Everything in your body is made up of these cells. So, and they're directed by proteins, which get kind of which find their marching orders from the DNA, those genes. So I know it's really it's kind of a really very, very shallow overview of what that process is, but that's all you really need to understand for the purposes of this class. Again, if you've taken a biology class, you probably know it in a lot more detail, like how do the proteins go and find and know what part of the gene to read, and then how do they know where to talk to a cell and things like that. You don't need to know all of that. Just know basically the molecule of DNA has a section on it that gives this protein an instruction to tell a cell what to do. When cells are all being told what to do, it tells our bodies in general what to do. So, what do genes do? So, with things like stress, exercise, nutrition, radiation, temperature, sleep, all these things can affect what genes then do, or how proteins read the gene. So, proteins are this complex molecule that get read by these, or sorry, DNA is this complex molecule, gets read by the proteins, and expresses genes, gives us our traits, controls cell functions. For a lot more detail than you need to know for this class, feel free to go to this link. It might help you understand a little better, even though it's definitely more than you need to know for the class. So the field of psychology looks at genetics and behavior. How do our genes influence the way that proteins are sending messages to our cells that then affects our behavior? So if our proteins are telling our cells and telling our bodies to produce more dopamine than other people or less dopamine than other people, that might be related to something like depression versus not, right? So we look at this in a couple different ways. Twin studies is one way, as well as adoption studies. So when looking at twins, you'll be comparing similarities between the twins. So with that, identical and fraternal twins you might look at differently. So identical twins should have the same genetic structure. So their genes are all the same. So looking at the things that are similar in their behavior, you might be able to find some evidence that some things are genetic versus what is behavioral in the environment. Similarly with fraternal twins. So it helps determine if any kind of behavioral trait or way people think, because remember, psychology is all about the ways people think, feel, and behave. So if these two twins are thinking in a similar kind of manner, like solving problems in the same way, if they're accessing memories the same way, maybe that's genetic, right? Or maybe it's not. Maybe it's both very different. So maybe that's something they've both learned out in the environment. So adoption studies do something similar. They'll compare the adopted child to their biological parents, if possible, as well as the adoptive parents, see what kinds of features might have been genetic, what features might have been from environment that they're in. So with that, a lot of these studies are correlational, looking at the correlations between behaviors and genetics. So remember, correlation does not mean causation. You can probably expect that to come up on this first exam as well as your final exam. So how do our genes get affected by the environment? There's a couple different ways. One of them is passive genotype environment correlations, and we'll describe those. Another one is evocative genotype environment correlations, and another is active genotype environment correlations, also called niche picking. So let's talk about all of those right now. So the passive genotype environment correlations, I think it's pretty easy to understand. Essentially, the environment that an organism, in our case, like a human, generally we're talking about a child here. So a human child is in an environment that's already supportive of their genetic predispositions. So they probably have the same or a very similar genetic makeup to their parents, if it's their biological parents, right? So let's say, just as an example, let's say the trait to read fast, like to speed read, let's say that's genetic. I don't think it actually is, just it's an example for the class. So let's say the trait to speed read is genetic. Well, if you had a couple parents who were both speed readers, they would probably have an environment with a lot of books. They might have a big library, lots of books on the walls, lots of books for the kids. So the kid has not done anything, the child has not done anything, but the environment is already supportive of that genetic predisposition. So that's the passive genotype environment correlation. The organism, that child, has not done anything. The environment already supports their genetic predisposition. So let's talk about evocative phenotype environment correlations. This is when the characteristics of that child, of that organism, elicit certain types of environments. So the way their own traits make other things happen in the environment, not necessarily intentionally. So a child who has the genetic tendency to be more friendly will probably elicit more friendliness from other children and then have more friends. So a child who genetically has the predisposition to be maybe a little more grumpy or uncooperative, they might have less friends. They might elicit different behaviors from other people. So this is evocative genotype environment correlations. So their environment will be changed because of their behavior, because of their genetics, in a way that then might support or create a feedback loop with that genetic tendency. Again, I don't know. I'm just using these examples. I don't think being grumpy is like a genetic trait. Otherwise, my genes change when I get more or less sleep, which we'll talk about epigenetics in a second. Active, I think that's funny because I know what's coming, but you're probably like, why is he laughing? Sounds very strange. And epigenetics is a little bit strange, but we'll talk about it. So active genotype environment correlations is similar, but in this case, the organism or the child is picking specific environments that are supportive of their genetic traits. So let's say a child like this one has a genetic tendency or predisposition to have more endurance, or to have more quick muscle fibers, or to really enjoy taking risks or something like that. Any kind of possible genetic trait. I'm just making these up as examples. So they might pick environments like going to playgrounds more, or picking a sport that's a little more active. Maybe they do parkour rather than golf or something like that. An outgoing child may put themselves in more clubs. So they are actively picking environments that are supportive of their genetics. So again, so we have the passive ones, which is when the environment is already ready-made for the organism, for the child to be supportive of their traits. Evocative, where the child's traits tend to elicit different things within the environment. And then active, or niche picking, where the child chooses specific environments that are supportive of it. Any questions about these, or examples that you have in your mind that you want to see, hey, does this fit more in this one or this one? All right, if you're just getting to the comments to type a comment, or slow at clicking the hand, that's totally okay. We can jump back to this if you have questions about it. All right, so you might see that these are bidirectional interactions, like we've talked about with a lot of psychology. A lot of things affect other things. These are bidirectional interactions between heredity and our environment. So our genes can affect our environment, and then our environment can then affect our genes, whether they're supported or not supported, things like that. So, for example, a baby might inherit genes. The prenatal development in, say, toxins might lead to certain genetic expression, or if they have healthy nutrition prenatally, that might lead to different gene expression. And then based on that, genes might activate or not based on that environment. So epigenetics is the maybe most dramatic example of our environment affecting our genetics. So epigenetics is about the changing of the molecular structure of the DNA strand in a way because of environmental inputs. So essentially, the environment can impact the structure of your DNA strand. And we'll talk about that over the next couple minutes. So if you're like, oh my goodness, how's that possible? We'll talk about it. So, but one thing I wanted to point out, just like this model of bidirectional interaction, a child might influence their parent, a parent might influence a child. child is influenced by school, school is influenced by that. This is another interesting thing, that our environment can also even affect our genetics, how our genes are expressed specifically. All right, so the book gives a couple examples of this. So the propensity for depression on the, I'm not actually going to read it, well, maybe I will, the 5-HTTLPR gene only activates when someone has a stressful life. So for someone whose life is more stressful, so let's take two twins, right? So twin A and twin B. Twin B ends up having a lot more stressful life. Maybe they're put in classes that are more difficult for them. They're in poverty. They are not able to find good jobs. They have a hard time eating. So they've got a really stressful life. So both of those twins have the same exact genetic structure, but twin B has this stressful life. That will actually change the way that molecule of DNA is basically wound. So the molecule is literally like curled up in a really complex way inside the body, and then the protein then goes and reads that chain of the molecule. I'm acting like my arm is a molecule of DNA. Visual examples help me. So for one person, maybe that's not at all twisted at all. So the protein comes and reads for twin A. It's not twisted at all. The protein comes, reads that part of the DNA, says, okay, no depression for this person. Awesome for them. But for twin B, maybe because of that stress, their DNA strand itself is twisted. The code is not changed. It's like A, C, T, G. It's still in the same order, but if it's twisted up more, like too twisted, then that protein comes to read. It might read it a little bit differently. Maybe it's missing one of those. Basically, because of the way the molecule is wound tightly or loosely, it's sending a different message to the cells. And that message then ends up with sending a message to, oh, twin B is going to suffer from depression. So that's one example. We're going to give a couple more. Histones, exactly. And you don't actually have to know what a histone is in a lot of detail, but that's a big part of the epigenetic process, is how histones are interacting with the strand of the molecule of DNA. So we'll watch a video that will bring up histones, but it won't go into them in too much detail. If you already know about them, awesome. Hopefully this will all make a little more sense to you then. All right, so remember what genes are doing is they're either creating proteins or proteins are reading the genes from the DNA, and that then sends the message. So the DNA strand itself, the code between these two twins is not different. It's just the way the histones are tightening, winding the gene or winding the DNA strand. Let's watch this and get a little more clear picture of it.
So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, clone probably has a tattoo on its face. And if clone had stayed in the circus, clone probably, professionally rides a unicycle over a tightrope, so clone is probably in pretty good shape. But then again, clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So at 50, you and clone would probably look pretty different from one another. But it turns out you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that other person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference. And if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely potentially changing the message of that paragraph. The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern, and that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life, but these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary, and it can change over time, especially when your body is going through a lot of changes, like, say, during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're going to have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're going to have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly, because it's true that a lot, even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know, it's such an unbelievable buzzkill. There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors a parent experiences can be passed down from generation to generation was sparked in the 1980s. And this happened when some scientists were looking at the birth and death records of some people who lived in 19th century Sweden. This is a weird place to find a genetic revolution, but there it is. It wasn't just any place in Sweden. It was in Norrbotten, which is the northernmost county in Sweden, which is literally in the Arctic Circle. And despite the fact that Norrbotten was literally the worst place you could possibly choose to live in Sweden, there were some people living there in the 1800s. And they were completely cut off from the rest of the world. Let me clarify. These people were isolated. Like, if they didn't have a good crop year, people died. No, it wasn't ideal, but, you know, they didn't ask me my opinion, so I'm not giving it. So anyway, these people subsisted entirely on what they grew and the animals that they raised. And like I said, sometimes they starved. But sometimes they had huge, bountiful years of plenty. And when it happened, people totally went ape crackers. I mean, of course they did, because they were so freaking hungry, and then all of a sudden there was all this damn food everywhere. Anyway, there was a public health specialist who was looking at the effects of the people who grew up in the really bad starving periods of time versus the people who grew up in the eat all you can at the smorgasbord years. And you might already be guessing what they found out. People who went from relatively slim pickings to feed your face until you have to barf and then do it all over again in a single season, those people died an average of 6 years sooner than their starved out counterparts. And you know what sucks? So do their kids. And so do their kids' kids. Boo epigenetics. So, of course, now we all know, and we're all going to stop doing unhealthy things starting today. Unless the damage is already done. The damage is almost certainly already done. But hey, epigenetics brings good tidings along with the bad ones. For instance, we now know that certain types of cancer are caused by misplaced epigenetic tags, and scientists are now developing drugs that can silence the bad genes that were supposed to be turned off in the first place. Additionally, until recently, we thought that genes were the end-all and be-all of who you got to be. They were your blueprint, and you couldn't escape them. This outlook is not just kind of depressing, it also leads to a yucky sort of social prejudice. Because when you look at data without considering social and epigenetic factors, it might look like people with less money are less intelligent. So just like 15 years ago, there were scientists saying things in public, mind you, like, some people just have good genes for intelligence, and it just so happens that the poor people don't. Oh, it's so sad. Too bad, poor people. But you have bad genes. Well, it turns out that that is not even a little bit true. Not only are there a huge variety of social factors that affect how well people do on intelligence tests, but a genetic trait is also not just a product of genes, it's also a product of environment. Any one person's genome is determined by any number of decisions made by any number of their ancestors. And right now, you are making decisions that are going to affect people who are alive long after you're dead. No pressure or anything. And so I'm glad you've watched this, because when you and your clone are in your 60s and you sit down, have lunch, and he brings his son and his son has a tattoo on his face, he did not inherit that from you. This is Hank Green for The Sci Show. We hope you learned something.
So epigenetics is one thing that I find just immensely fascinating. Had I learned about it at the stage you are in your programs, I might have gone into more detail on epigenetics in my studies. So this is one reason I like to emphasize it to students, because it's something that is exciting to me. Hopefully it's exciting to you. If some of that didn't make sense, if some of the words didn't make sense, that's totally fine. But hopefully you got the basic idea that our environment, the things that we say put into our body with our food, with the environment, pollution, things like that, can affect the ways that genes are expressed. Again, it doesn't affect the genetic code itself. So if you cloned yourself, your clone that is in a different environment would still have the same genetic code. If you analyze the genes, it would all be in the same order. But those methyl groups and how the genes are wrapped around those proteins called histones is going to be slightly different and lead to different proteins activating different things in cells in different ways. So our environment can affect how our genes are expressed is probably the most accurate short way to say that, because it's not actually altering the genetic code itself, it's altering how those genes are then read and expressed. But pretty cool. There is another example video about Pokemon. So if you've ever played Pokemon or watched any Pokemon cartoon, it might be relevant. We're not going to watch it in class. We don't have enough time today. It just gives another couple examples. One of the examples in the video is actually of a mouse, where if they are given too high of cholesterol, their coloring changes. So as well as their size. So too high fat and cholesterol, the mouse changes from a brown to essentially a blonde mouse, and it's a lot larger. in that kind of case. And that's because of epigenetics. It gives another couple examples. One of them that might be a little more familiar to you is actually the honeybee. So a honeybee can actually be either a queen honeybee or a worker honeybee, but depending on the diet that it's given, will activate different genes based on those epigenetics. So any of the bees could become the queen bee if they're given a specific kind of food called royal jelly. So pretty interesting that just with a diet of royal jelly versus the other normal foods that a bee larva get, a bee will then be a queen bee versus just a normal worker bee. And that's epigenetics. So it's another good video with another couple of good examples. The basic idea is that our environment can affect our genetics, and some of those changes to our epigenome with those methyl groups, those tags they were talking about, can be passed on. So not everything's going to be passed on to a next generation, but some of those things can be. And the more it's investigated, it seems like a lot more than we thought previously can be passed on. So something I think is interesting. Questions that you have for clarifying this. You can also do, say, a question paper on this. Feel free to raise your hand or type in a question. So I think this is a really interesting concept when we think of how our environment is affecting us. So we saw this model is a developmental model of how people change through time. Another way people change through time is by their environment affecting their genes. Well, their genes expression. Should be clear about that. Try to not mess it up. So things in our environment might lead us to maybe more positive outcomes or negative outcomes. So developmental cascades is the kind of model thinking, the model around that kind of thinking, that as we go through life, different things can happen that can then affect how we then respond to them and how we respond to things later on in a cascading effect. Something happens with our epigenome that leads us to positive outcomes and more positive outcomes and more positive outcomes. That would be really great, man. That's one of the things that we want to do as applied scientists, try and help people get those more positive outcomes. All right, so heredity can be influenced by our environment through those correlations or as well as through epigenetics. So before we jump into chapter 3, let's take a quick break. We've had a lot of information on question papers, exams, and now we finished up chapter 2. While we're on this, let's say 5-minute break, feel free to type any questions in as well. You can send me emails after class too. But we'll get into chapter 3 on sensation and perception. My clock here says it is 8:12, so let's be back at 8:17, and well, it's 8:17, and go through chapter 3. All right. Hopefully everyone is back and can see me and hear me now. Let's jump into chapter 3. During my break, my cats were asking me why I hadn't given them food yet. Well, they're still asking. They want their food. All right, so chapter 3 is on sensation and perception. So, what is sensation and what is perception? Sensation is the signals, the information that we get from our sensory receptors. So, eyes, the light coming in through our eyes, the sound coming through our ears, that is the sensation itself. Perception is then, what does that mean? So what does the light coming into my eyes, what does that mean? Well, I can see on my little screen here, or this wall, I can see this teal color. So I'm getting this light message. What is that? That's the color teal. I'm getting other wavelengths of light. So the sensation is a different wavelength of light over here with some whites and purples. Over here, I've got some blacks and blues. You might not be able to see that one in yours, but there's some golds, greens, and blacks in this one, and then some reds and blacks here. So the sensation of those is just different wavelengths of light. That is the sensation. The perception then is, oh, that's a painting. That's a painting with red and black. So then I perceive that. What are we seeing? What is the shape? What's the color? What's the pattern? That kind of thing. As well for auditory perception. So the sensation is the audio signals, the sound waves that vibrate our eardrums, and then those hair cells. Hopefully you've reviewed this already in chapter 3. All right, so this is related to bottom-up and top-down processing. Bottom-up is the sensory receptors registering information, sending it up to the brain. So eyes getting information, sending it to the brain. Your skin getting pressure information, sending that up to the brain. And top down is then our cognitive processing. So we get that message to our brain. Now what do we do with that? Well, I have a sense of something on my leg. What is that? Oh, is that a spider or is that a feather? I might look down and swat it away. Luckily, it was just a feather. Sorry to trigger anyone about spiders. So with that being said, what do you see in this picture? So the picture I have on my screen here, what do you see? Feel free just to type it in the comments. Hopefully we get a couple responses. The answer might be nothing, might just be like black dots, right? So what is in this picture? Oh, my mic has been off that whole time. Sorry about that. So we've got a couple of people who see trees and fields, lips and hair, a lake, a pond, things like that. So there's some shapes over here. Maybe people see a pond. I can see how that would be lips as well. A field. Some of these look like leaves. Someone said up here in the upper left, that looks like a tree. Yeah, so I see a tree right here. So essentially these white kind of vertical lines are the tree going up. This is the base beneath the tree, whether it's just shade for me or like dirt around a tree. And then I actually see a dog right here. So its nose is towards the ground, sniffing something. It's a Dalmatian. Here's where its back is, and then a leg, another leg, another leg. Then its fourth leg is kind of obscured. And this is like a path that it's on the edge of a path. So that's what I see here. Other people see different things. So what does that tell us? Well, what's our sensation of this? Hopefully maybe now some of you see the dog and maybe you still don't, and that's okay. The point of this picture is to be a little bit, you can kind of interpret it in different ways. So with this, what is the sensation that we're getting from this picture? Well, we're getting the light information, right? We're getting some whites, some blacks, and that sensation. The perception part is what kind of patterns now am I going to draw from this? So that's the top-down processing. What is the pattern? Am I going to see lips? Am I going to see a lake? Am I going to see a dog? So that is then our brain perceiving what this is. So I like a lot of different visual, what's the word I'm trying to look for? Optical illusions and visual tricks use sensation and perception in that way. Make you perceive something because of your sensations that's a little unusual. All right, so that bottom-up processing is that sensation, the light coming in, the blacks, the whites. The top-down processing is perceiving, oh, is this a dog? Is this a lake? Is this lips? Is this a tree? Those kinds of things. So what do we get these senses from? What do we get those sensations from? From sensory receptors. So sensory receptors are specialized cells, specifically a specialized neuron. So each of these is a drawn example of a different kind of neuron that select a specific kind of energy and then send that information to the brain. So for vision, we have rods and cones. So this is an example of a cone. This is an example of a rod. From those, we get the light information, and then the neuron then has an axon with some axon branches that eventually goes to the optic nerve, then up to our brain. For hearing, this is an example of a hair cell. For touch, we have different pressure sensitive as well as pressure and temperature sensitive cells for touch. Smell and taste are pretty similar in a lot of ways. So we are getting these sense chemicals and then send a neural signal up their axon. So they're just a specialized type of neuron. All right, so we get three distinct kind of energies. One of them is photoreception. You probably guess that's about light and sight. Mechanoreception, and that's like mechanical, you can think of it that way. So that's like touch. And then chemoreception, which is chemicals. So photoreception, rods and cones, they're lining the backs of the insides of your eyes. Interesting, well, we'll go into more detail in the eye. But this is how we get our perception of sight. We get the light. Mechanoreception, hair cells, so your ears. So this earpiece that I have in is actually using mechanoreception, pressure changes of hair cells. So the sound wave, then, well, we'll talk about the ear. And chemoreception is those chemicals that you can taste or smell. These neurons get activated and then send a message with a chemical. So pretty neat. All right, so absolute threshold is for these sensory receptors, for these specific neurons, absolute threshold is the minimum amount of a stimulus that is needed to then send a signal that you've now detected this thing. So the minimum amount of salt that has to be there for you to detect it, the minimum amount of light that has to be there for you to detect it, that's the absolute threshold. So your very minimum threshold is the absolute threshold. So on a clear night, when there's not a ton of smoke in the air from fires, for example, and there's not other light pollution, the eye can see a candle flame from about 17 miles away. So that is the absolute threshold. You could perceive well, you could sense and then perceive that little pinpoint of light. Touch, this example is a little funny, the wing of a bee falling on your cheek from about a centimeter away. Your mechanoreceptors and different parts of your skin are more sensitive and less sensitive. So you're more sensitive on, say, your fingertips for mechanoreception than, say, your toes for mechanoreception. For taste, you can, unless you're my children, you can taste about 1 teaspoon of sugar dissolved in 2 gallons of water. If you're my children, you can't taste the sugar at all until it's like 2 or 3 cups in those 2 gallons of water. So I'm sure that's not actually the case, but usually they're like, put more of the Gatorade powder in the juice or whatever. So smell, you should be able to smell about 1 drop of perfume in a 3-room house, and then the ticking of a watch in a room about 6 meters away. So that is the absolute threshold, the minimum that humans are essentially capable of sensing. So difference threshold is a different thing. So this is how much energy is required for us then to sense a difference in the level. So this is called the just noticeable difference. So I've got a picture of someone shooting at a shooting range here because this is a really good example. When you get to that high levels of energy, the difference threshold is actually really big. But when you're at a really low level of energy, like whispering for audio, the difference threshold is a lot smaller. So if you do a little whisper, I don't think a whisper would actually come through the microphone very well, so I'm not going to try to whisper in the microphone. You've probably experienced whispers in your life. So if someone whispers a very, very little bit and then a little bit more, you can tell that difference. Even if it's, let's just make up a number. Let's say it's 10 decibels louder. You can tell that difference from a whisper to a little bit louder whisper. But let's say you have a rifle that you're shooting at a shooting range. Generally, the shorter your rifle barrel, the louder the sound is going to be. So let's say you have a standard like 16-inch rifle barrel. It's a pretty loud sound. You should probably be wearing hearing protection then versus someone who has, let's say, a 7.5-inch rifle barrel. That's going to be a lot louder sound decibel wise. If you had a decibel meter, it might tell you, oh, well, now it's 200 decibels louder. But to your ear by itself, you might not actually notice that difference. It's just going to be really loud and really loud. You might not be able to tell that one is louder than the other in that same room. So even though that's like 200 decibels louder or something like that, you might not detect that difference, whereas you might detect at the whisper levels that 10 decibel difference. So that's what we're talking about, just noticeable difference or the difference threshold. Oh no, I left my tea over on that table over there. That's so sad. So the just noticeable difference does increase with the strength of the energy of that thing. So if you do have 2 cups of sugar in your gallon of juice, and then you add another tablespoon of sugar, you might not notice that difference. If you have only 1 tablespoon in that gallon of juice, and then you add a 2nd tablespoon, you might detect that difference, but when it's so high, you might not detect it. All right, so another important thing with sensation and perception, very important for a lot of things like memory as well, is our attention. Attention is simply the focusing of our mental resources on specific information. Hopefully you're attending to the class, you're attending to the lecture, you're attending to the book as you're reading through it, you're attending to the videos. So as you focus your mental resources, you will be able to improve your cognitive processing. You'll be able to tie things to memories better. You'll be able to understand it better. You'll be able to think up questions about things. Again, if you have any questions, feel free to raise your hand. I know it's a little more difficult in this kind of setting for me to really gauge if you're understanding, if things are clear, if you're all really confused. Whereas in a classroom setting, I might see your faces that everybody looks confused versus here, I don't really get that same sense. So definitely let me know. And if you feel uncomfortable mentioning it in this kind of setting, email me afterwards. That's fine as well. So we do understand and perceive things better when we attend to them, when we focus our attention. So selective attention is when you select a specific thing to attend to. So focusing on something specific, ignoring things around. So let's say you are at a dinner party with friends and you're having a conversation with one person. You might focus on that conversation and ignore the conversations that are going on around you. This can also be called the cocktail party effect, the ability to focus on that one conversation. You might have also had the problem where you're having a conversation, but then this conversation over here is actually more interesting. You start focusing on that one, and then you're no longer attending to the conversation over here anymore. So why do we choose to attend to things? What moves our attention in different ways? Well, we've evolved to attend to novel stimuli, to things that are new, different, unusual, whether it's size, color, movement. So as an example, in my tiny little video screen, I just saw like a bright white light. That was my wife's laughing at me now. That was the freezer being opened over there. And so I then attended, my attention went to that. So I attended to that change in the environment. And we evolved that to be safe from like saber-toothed tigers jumping out and eating us so that we could attend to those changes in the environment and survive, right? So inattention blindness is one thing that is relevant here. If there's something that is very similar in the environment, you might not notice it. So let's watch this video. Try to follow along with the directions. I think it asks you to count the number of passes one of the teams makes with each other. So this is an exercise on inattention blindness. What am I doing? I accidentally pushed a button on my monitor and it wanted to do something different. All right, there we go.
This is an awareness test. How many passes does the team in white make?
Go!
The answer is 13. But did you see the moonwalking bear?
So raise your hand or make a comment if you did count the 13 passes. Or if you got like 12 or 13 or 14, if you got pretty close. So we've got 4 raised hands. We've got 11. Okay, so it looks like we've got probably about 7 or 8 students who got around the right number of passes. So of those of you who did get the right number of passes or pretty close, so 9 or over, did any of you see the moonwalking bear? Those of you who got around the right number of passes, did any of you see the moonwalking bear? Oh, it looks like we've got 1 celestial. We've got Nicholas, Carter. All right. So a lot of no's though. Yeah. So if you're attending to something, to the people passing, like I asked you to do, then that's what your attention is going to be on. There was a change in the environment. There was a person in a bear suit moonwalking through the group. They were in it was basically a black suit. It kind of looked like a bear. So if you're focusing on the people in white, you might miss them. That's inattention blindness. So this video will then go back, kind of rewind and show you that. So hopefully that second time you did see them, just right here in the very middle. So kind of a funny exercise to trick your sensation and perception and show you what inattention blindness can do. So while you're focusing on the people in white passing a ball, you're not paying attention to the very subtle change in the environment. If that person in the bear suit, if it was a rainbow bear, you'd probably kind of hoping for a real bear. That might be a little more easy to detect since it would be such a different shape. Or a rainbow bear or something like that would be probably a lot more easy to detect because the change is more dramatic. It would be a more novel stimuli. So with that, this can happen to us just in our day-to-day life. Yeah, so sensation is you probably got all of the light. Oh, you're looking for a teddy bear or something? Yeah, there was a full-sized human. Here, I'll go back to the video really quickly. Well, at least the shot. So this is that person right there, and they're dancing. So they're in a bear costume. So the sensation is, if we're watching that, probably every single one of us got the light energies, got the sensation of the person in the bear costume, as well as all the people passing the ball. But we didn't perceive it necessarily. One of the reasons is attention. So attention is a big part of this whole process. All right, so with that, let's break into some small groups for just a couple minutes. So let's come back all together at 8:46. But jump into groups again. Here, I'll actually show you the screen to describe this. So you should see what I see here. So here we've got chat, things like that. Close out of this video. Actually, I might not be able to show you. You don't actually have to leave the video. You can. Now I've forgotten how to do it. Go to things like activities, and hopefully the video is still going for you. In general, you can go to your teams. That's where you go. Sorry. So go to your teams, choose the team that lines up with your last name. So A through E, F through L, M through P. So my name starts with R, so I will have to run through the whole alphabet to remind myself, am I in this group or this group? So jump into your group for the next couple minutes. So we'll come back at 8:40. Let's come back at 8:47. So talk about, in your small groups, how is this related to driving, to texting and driving? How is inattention blindness? How is attention? How is sensation and perception? How is this related to texting and driving, specifically this inattention blindness? All right. Hopefully all the groups are heading back to this main group now. I heard some good conversations in those groups. It seemed like enough time for a few groups and not enough for a few. Some technical difficulties made it difficult, and some people were not able to do video and audio. Some people just were able to do chats. But does anyone want to raise their hand to share some of the main points that they talked about? How what we're talking about, inattention blindness, sensation, and perception are related to texting and driving? Go ahead, Don. You've got your hand raised. What did your group talk about?
I mean, we just talked about how, I mean, like we were saying, when your attention's on something else, it's really hard to focus, and you can't necessarily be focused on both, because even when your attention's taken away for just, a few seconds, in those few seconds, someone could step on their brakes in front of you, and yeah, their brake lights like light up and everything, but I mean, I know from experience that I don't always see that until I look back away from my phone. And it's I feel like a lot of us nowadays have like those phone mounts on our dashboards on our car, and so even or in our car, and so even when we're not necessarily texting, even just like getting notifications or something, it just lights up right in our face, like pointed at us. And so even just glancing over for a few minutes, and then of course, if it is something that we want to respond to, it's really hard to like resist that because it's right there in your face,
Yeah, definitely. Really great points and great connections to sensation and perception. You're absolutely right with all of that. So, with that, tying it into last week's chapter on neurology, if you've been driving for a while, which I assume most people in the class have been driving for at least a few years or even many years, if you've been driving a little while, it's now probably become kind of an easy process. It's kind of a habitual thing. You might be always driving the same route to, say, work or school or home. So you kind of get in a habit. You're used to your own car. You're used to where things are, the roads that are around. So we start to pay less and less attention. And when we're doing that, we're being less attentive to the environment. So if something happens like a child runs out into a street, someone slams on their brakes, it's harder then because we're not attending to that environment to do that. So basically our neurons have learned how to drive our car around that same path that we always take. So it makes it even additionally more hard with this sensation and perception. And that's just a feature of our biology. Our sensations, our senses, our perceptions are trying to attune themselves to new and novel stimuli. So our phone is literally designed, just like you said, to give us new and novel stimuli to vibrate or make sounds. And we also have associations, whether it's positive or negative, with those things. For example, I think a lot of millennials as well. I'm not sure about Gen Z. Some Gen Z people let me know. When I hear a phone call, that's not as necessarily as positive an association as a text, because I might have to be talking to someone versus them texting me or getting an email or something like that. So I don't think that's unique to me. Maybe it is. Oh well. But yep. So very good. So texting and driving, if our attention is on that text, on that phone, it's going to make it a lot more difficult to attend to other things within the environment. All right. So continuing on, we also have sensory adaptation. Oh, and I lost all the chats. Where did they go? Okay, got them back. So our senses can also adapt to things. This is the change in the responsiveness of our sensory system based on the level of environmental stimulation. You might have experienced this when stepping into, say, a hot tub or a really cold pool or lake or something. You can adapt to that sense. So at first it might be really, really hot or really, really cold, but then when you're in that water for a little while, you get used to it. You're adapted to it. Same thing in a dark room. You might walk into a really dark room and not be able to see anything, but then your senses adapt. They change their responsiveness to be able to do a better job seeing and sensing in that environment. Another example is you're probably not registering the feeling of your clothes on your own body while you're in class or driving or just doing something generally. But now that I mentioned it, now that I'm mentioning it, I literally feel my collar and I'm like, oh, there's a collar there. That's kind of annoying to me. But before that, I didn't register it. I had adapted those neurons, like the pressure sensitive, those mechanoreceptive neurons on my neck. They were no longer sending a signal, or the signal was so small that I was completely not even aware of the collar on my shirt, as an example. So for our visual system, let's talk about the visual system and auditory system in detail before we get to another couple points in sensation and perception. So light is simply an well, maybe not simply, but it's an electromagnetic wave, an electromagnetic energy that can be described as waves. So a wavelength is the distance between the peaks and troughs of that, and we perceive that as a hue, as a color. So you probably perceive a couple different hues of gummy bears right there. Amplitude is the height of the wave, so how big the wave is, and we perceive that as brightness. So something that's smaller like this we would perceive as more dim, than this we would perceive as more bright. Purity is being the same wavelengths or versus a mix of wavelengths. So this is a pretty pure hue, so let's say that's the hue purple. Actually, that's probably not a good example because purple would actually be multiple wavelengths put together. Let's say our receptors are red, green, and blue, and I forget if humans have yellow receptors. We'll probably get to that in a minute. So let's say this is a blue. This wavelength here. This is a pure blue color versus this is, let's say this is purple. So it's multiple waves. They're combining themselves, not entirely pure. And we perceive that then as purple. You can also see perceived as saturation or the richness of the color. So this has a lot of white in it versus this has some more blacks in it. So humans only see a small portion of light energy. So visible light is this segment right here. It's from about 750 what is that, nanometers to 400 nanometers. So the shorter the wavelength, the closer it is to violet and ultraviolet. The longer the wavelength, the closer to red and infrared. So on this side of visible light, there's other light energy, infrared, radar, microwave, radio, and then on the upper end that we can't see is ultraviolet, X-rays, gamma rays. So flies, for example, can see more ultraviolet than we can. Actually, most birds can as well. If I'm not mistaken, dogs can see further into the infrared spectrum than humans can. But I think their visual system is limited in they can't see as high of hues as humans can. So kind of interesting. So the structure of the eye is pretty similar to a camera. If you're familiar with a camera, our own eye is very similar. So we have a pupil that lets light in, that black part. It passes through a lens. So we literally have a lens that can stretch and contract. Well, assuming it's healthy, it can stretch and contract. As we age, a lot of time we might lose that ability to stretch, and that's why it can be difficult to focus on things further away and closer to us as we age. The light is flipped in our eye. Yes, I am recording. It should still be recording, even though I accidentally hung up during our group breaks. It should have recorded this whole thing. So, yep, you can definitely re-review the lecture recordings. It hasn't been saving them on Microsoft Teams. It's supposed to, so that you can just come back here and click on it. It hasn't been saving there, but I have been putting it on YouTube and then emailing it to students. So you can get it there as well. I'll post it on Canvas. So find it on Canvas or in an email. So, but it definitely will be on Canvas. So yeah, eye is very similar to a camera. The sclera is the white part. It gives you kind of the structure or shape of the eye. You can think of it kind of as your eye skeleton. So it's the hard part that keeps it round. The iris is the colored part. It's actually muscles that control how much light gets in and out of the pupil. So you could think of it like a camera's aperture. So a camera's aperture, usually it's a bunch of gears that kind of spin together to have a smaller opening versus a larger opening, like that, kind of. So that's the aperture. That's what our iris does. The pupil is where the light passes through. The cornea is a protective layer, just a UV shield on a lens of a camera. That's what our cornea basically does. And then the lens is that flexible disc that changes our focus. So there you go. Then back here at the back on the retina are our rods and cones. We have a lot of rods in our fovea, and that is where our sharpest vision is. So when you look at something specifically in the center of your vision, that is going to the fovea. The things on the outside in your peripheral vision are hitting this part of the eye, and they're not in as much detail actually. So your fovea is the most sharp point of your vision. Then around that, you can still see things, but there's not as many rods and cones. So it's like the very center of your vision is HD, but then the rest of it is not HD. So the retina is where the rods and cones are housed. Here is a microscopic picture of rods and cones together. So rods are receptor cells that are very sensitive to dim light, but they are not useful for color vision. So for black and white, for the brightness and the intensity of brightness of things, we have about 120 million rods in our eyes, and then cones are receptor cells for colors. We have specific cones for specific colors actually. And it needs a lot more high intensity light to see color, which is why when you're in a really dark room, things generally just look kind of like black, white, gray, because you might not have enough reception or you might not have enough energy for your cones to be picking up any color information. We have about 6 million cones, and we have multiple different cones for multiple different colors. One thing I find interesting is that looks like we've got a good comment here from Parker. Okay, that's a really good question, tying in a couple different things. So if you don't have the chat box open, I'll read this for you. Parker says, I tore and detached my retina when I was 18, not due to any accident. His grandfather has also had surgeries on both of his retina. The theory was that this might be genetics, but he asked, would this be epigenetics? So that's a good question. And it would be really hard, I think, in this specific kind of case to say that that specifically was epigenetics versus just genetics. If it's happened to many other people in your family, I would say it's probably just genetics. But if it's happening to a couple specific individuals directly in a line, so say you, your grandfather, your own children, their children, it might possibly be epigenetics. It could, if it's something like you and your grandfather always had the same kind of diet, that might come out epigenetically. So possibly, yeah. I would suspect probably more genetics than epigenetics. But again, there's a lot of things that we're still learning about epigenetics, in that something your grandfather or even your grandfather's grandfather did might then have led it to be more likely that people in your family line might have a detached retina. So, yeah. Why do people see the same color differently? That's a good question too. So that's all about how it can be a lot of different reasons. Yeah, especially if it's happening to you when really young. Parker's story is probably related to genetics. So Lindsay asks, why do people see the same color differently? So that's a good question. So here in the back of our eye, in the retina, this is the very back of your eye. And then right here We've got ganglion cells. These are bipolar cells, and then these are the rods and cones. So your rods and cones are actually behind a couple layers of other neurons, which I just find fascinating that we evolved this way, that the rods and cones, the actual light sensitive parts, are being occluded by other cells in front of it. So the light is coming this way where my mouse is hitting, so it has to pass through these ganglion cells, pass through these bipolar cells, and then hit the rods and cones. So why do people see the same colors differently? Could be a couple different reasons, or all of them put together. So if one person, people who are colorblind, for example, one of the most common color blindness is red-green color blindness. And a lot of those people, not all of them, but for many of them, it's because one of their cones that detects rods and cones does not actually function correctly. So they're not actually getting the signal of that. Similarly, people might have certain rods that are more active or more sensitive than others. So that could be part of it, that the rods and cones themselves, well, in this case, probably cones because they're the color sensitive ones. So that it's the cells themselves. They're either not sending the information, not sending it as strong, not sending it as often. And so that's why they might perceive it slightly differently. It can also happen. So what happens is these rods and cones, they get the light, then they kind of compress it. You can think of it like an image being compressed. So a JPEG is a compressed image. A RAW file for an image is all the pixel data all completely together. But if you save it as a JPEG, if you don't know anything about photos and files, this example might not help at all, so I'm sorry. But essentially, when you take a picture with a digital camera, you get data from every single pixel of the sensor. When you save a RAW file, the RAW file is a pretty big file and it has every single pixel of data. You can think of every single pixel of data as the rods and cones, and then it compresses it into a JPEG with these bipolar cells. It basically takes a cluster of rods and cones around it and clusters it. It says, hey, all of these are kind of seeing about the same brightness of green. And so it sends just a new message from this bipolar cell of there's green over here, or there's a teal wall, right? So it helps save energy. It helps that message go faster. It helps fewer messages be sent to the brain. And then even further, it will compress it down again to these ganglion cells, which then go to the actual optic nerve to the brain. So in that process of compressing, it might compress it in a certain way that, or someone might be more likely to compress certain types of colors, maybe because of the rods, or sorry, the cones, so that they're perceiving a certain color slightly different than someone else. So through that compression process, you might then have a change in how you're then perceiving a color, or it might be from the cones themselves. So kind of a long answer to Lindsay's, but it also is really relevant to the structure and function of the eye. And then Don Mira, you said experiences, and I'm not quite sure what you mean by experiences. Can you expound on that while I continue on in the slides? If I already answered your question, no problem either. All right. As we think about that, we actually have a blind spot in our eyes right here where the optic nerves. Oh, yeah. So husband only sees pink, but not rose. So for that, for red hues, I would suspect there's probably a problem with, again, I'm not a medical doctor, like you'd actually have to get this analyzed and assessed. But because it's specifically that color, I would suspect it's a problem with the cones that detect red, the wavelength of red, would be what I would suspect. Not rust color, just red. Yeah, so if it's around that same hue of red, I would suspect that it's because of just that specific kind of cone. I didn't hear that, actually. So Don Grover says, I've heard that women naturally see reds better than men. I haven't heard that one, but maybe a good thing if you think you've heard that, it could be a good thing to write a question paper on. So say, hey, in your question paper, you might say, I was really interested when we talked about vision and sensation and perception, and I had heard that women see reds better than men. I wondered if that was true. Find a research article on that. Find maybe a website that's relevant about it. But yeah, I hadn't heard that. That is really interesting. So I would be interested to read a question paper about that topic. Then I could use it as an example in the class later on. Yeah, good questions though. One interesting thing is that every single person has a blind spot in each of their eyes. This area where we have our optic nerve going out the back of our eye has no light sensitive neurons. So we all have a little blind spot in each of our eyes. So with your screen, depending on the size of screen that you're looking at, this is actually a blind spot test. So as you look at the red dot, you can close your left eye. So if you're looking at the dot on the left, close your left eye. So I'm looking at the red dot with my right eye, and if I move to right here, I mean, you don't really have a sense of what that is because you're watching me on a screen. I'm about 3 feet from my screen, and the green dot has now disappeared from my right eye. So again, my right eye is the only one that's open. I'm looking at the red dot, and the green dot has disappeared. I literally cannot see it. All that my brain is perceiving is white, the red dot and then white. But if I move a little closer, I start to see it again. It's like it reappears in my peripheral vision. Similarly, if I close my right eye, and I'm about that same distance, about 3 feet from my screen, the red dot disappears. So my left eye is open on the green dot, and the red dot has now disappeared. Because that dot, the light from it is going directly onto where my optic nerve is. And so that's why I can't see those dots. You can do this with your fingers as well. Since you're looking at a screen, it's probably a little easier to like move forward and back to find that where it disappears. So again, if you're looking at the dot on the right, close your right eye. If you're looking at the dot on the left, the red dot, close your left eye. Move closer and further back. At some point, the other dot will just disappear. It's pretty neat. You can do that with your fingers as well. So just hold them like out in front of your face. This probably looks really strange on that camera, but if you close one of your eyes and bring it closer or further away, one of your fingertips will disappear from your perception because you're literally not sensing that light. So pretty neat. Also relevant for things like street signs on the side of the road. When we're able to use both of our eyes, so if you have full use of both of your eyes, your vision from your other eye will cover that blind spot. So we've evolved to have 2 eyes facing forward that will cover up that blind spot. All right. So visual processing, we do have feature detectors within our neurology. We have specialized neurons in the brain that respond to specific features of stimuli. And this helps us be a little quicker even at processing visual stimuli. So we have sets of neurons, maybe even specific neurons, but clusters of neurons that respond to specific shapes. So one that's very common for that people generally have is the shape of faces and the shape of human silhouettes. So this is why so many people see faces of people in pieces of bread or mountainsides or clouds, is because we have these feature detectors, these clusters of neurons that activate when it sees a shape similar to a human silhouette or a human face. So size, we have feature detectors that detect things that are really big or really small. We have feature detectors that can detect specific colors, like red, for example, or movement as well. And the brain learns these patterns, learns like the shape of faces, learns the shape of human silhouettes, like what does a head and a shoulder kind of look like. through experience. So as we're born and we start seeing things, our brain starts to pick up on what are important shapes and create these feature detectors within our neurons. The neurons start to connect with each other. Genetically, we have the capacity for those neurons to connect up, but we're not inborn with the automatic feature detector of detecting faces. Parallel processing is essentially that we see with with our binocular vision, we see all aspects of an image at once. We see size, shape, color, movement, and then we sense that, then we perceive it. Trichromatic color theory is essentially the idea that color perception is produced by 3 different types of cones: the red sensitive ones, which we've talked about, blue sensitive cones, and green sensitive cones. So similar to how early TVs had, actually current TVs too, have red, green, and blue light that they put at different intensities to make all different colors, our cones work basically the same way, just in reverse. So, yeah. So with that, the most common color blindness is red-green color blindness. And so that, oh, I was wrong earlier. I said it was the red cones. It's actually the green cones that are malfunctioning. And so it makes it very difficult to distinguish green from certain blues and reds. So in these images here, the one on the left has a little 8 in red inside green dots. On the right, there's a green 5 inside kind of reddish orange dots. So if you don't see one of those numbers, you might have a little bit of color blindness. You might have already known that you had some color blindness, but on the one on the left, there's an 8. The one on the right, there is a 5. So with that, visual perception is also not just a sensation, it's the perceiving, how our brain puts things together. We put things together with shape. We use different Gestalt principles of closure, proximity, and similarity. So things that are similar, things that are close together, we tend to associate them with each other. So if you look at this on the screen, you might say, oh, that's a triangle. Well, not really. Like there's this break there. It's not actually a triangle, right? But we kind of close that up on our own. All these random kind of greeny, bluey, teal shapes that kind of looks like a horse or maybe a, I don't know, what is that animal? Some kind of animal. But in reality, it's just a bunch of random shapes, right? But we put it together. We perceive it as a whole. These dots here on B, we perceive, or you probably perceive these as 4 different lines, when in reality, they're just random dots, right? But we perceive them together. They're close together, we put them together. If you look at these, if your screen's big enough, you might be able to see that there's circles and squares on these red big boxes. One, we put these boxes together, but we also might delineate the lines because there's circles in a line and squares in a line. So we perceive visual things as well. So it's both bottom up and a top down process. Depth perception also comes from your 2 eyes being at slightly different angles to things. This is why VR headsets work. They put a different screen in front of both of your eyes. You get a slightly different picture. It gives you the sense that something's further or closer. We also perceive different clues by things that are overlapping. We learn that, well, these paintbrushes are overlapping that child in the background. So the paintbrushes are probably closer to us. We've learned that paintbrushes generally aren't as big as humans. So probably the paintbrushes are really close and the humans further away. So we learn that through experience in our environment, through practicing and using our visual senses. Shading helps, so things that are darker. So in the background here, you can see back towards my kitchen, it's darker there. Up here, it's brighter. So it gives you an idea of where light is coming from, things like that. Texture gradients. This picture here kind of looks like it's an angled plane, like going away from you kind of thing. Motion perception. We have a lot of feature detectors to detect motion because it's really important for survival. So this is really good feature detector to have for surviving a tiger attack or surviving something like driving to and from work. Perceptual constancy. So this is our perception that things can move and change. So even though the images we see may be changing, the objects themselves are not changing. So this person in these two different pictures, this person has not now morphed into a person that's facing that way, right? They just have now turned their head. The object themselves did not change. It sounds terrible to call a human an object, but maybe I should put a different example than a human here. But we know, because of experience, right, that people can move their heads and look different ways, that people don't just morph into being a different way. So with that, we know things stay about the same size, about the same shape, about the same color. So when we see changes in that, it draws our attention. So maybe that motorcycle that looks really small and then it looks bigger, well, that's probably because it's coming closer to us. And that's actually harder to detect than, say, a big truck that's far away and looks small and then it's getting really big because it's getting closer, because a motorcycle is a smaller vehicle anyway. This is a useful video about visual perception, but I don't think we have time to watch it in class today. It's kind of a review of a lot of things that we've talked about already. So feel free to watch that if you still want a couple more examples. I know we did skip a couple of the points here about visual perception. I did want to get to some of these illusions. So if you stare at any one of the circles, the white circles, it will look clear, and it may look like the other circles around it are blinking. So I'm looking at this circle, for example, right now, and it's just fine, but as I kind of wiggle my head around, it looks like there are black dots blinking in all these. So our brain is perceiving, trying to fill in those spaces, and it gives you that blinking effect. This picture is just a picture. It's not a GIF, it's not a video, but depending on where you look at it, different aspects of the picture might be moving, look like you might perceive it as moving or rotating. These lines are actually, the orange lines are actually the same size, but because of the principles of figure ground, it seems like this one is actually longer than this one, when in reality they are the same size. We might recognize some patterns here. You might pick out a circle, 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 a triangle here and a triangle here. So all of these yellows, so if you took a, well, assuming your monitor, your screen is giving you correct colors, all of these yellows are actually putting out the same exact yellow wavelength of light, but you perceive it differently against different backgrounds. So I perceive this yellow as very different from this yellow, but this yellow for me is pretty similar to this yellow. So here these oranges, well, the orange dots are actually the same size, but we perceive it as this one being larger than this one. And this is one of my favorites, is the A and B, both those squares are actually the exact same shade of gray. So what we perceive, we've learned that things in shadow are darker, so we then perceive it, we lighten it in our own brain. But if you connect these, you can see it is actually the exact same shade of gray. So we'll go back and forth a couple times. It doesn't ever change that A and B, they stay the exact same shade, but we perceive it differently when it's connected versus not. Yeah, I agree. That one is pretty crazy to me. One of my favorites. All right, so that's the visual system. It's one of the most used systems in our body, so I do want to focus on that. I also really like the auditory system, so we'll spend a little more time on that. We won't spend as much time on mechanoreception for touch or chemoreception for taste. But the auditory system, we get our information from auditory senses from the wavelengths of sound. So the wavelength determines the sound frequency, so its highness or lowness. And some wavelengths are too high or low to hear for humans, similar to how wavelengths for light are, some of them are too high and low for humans as well. But other animals like dogs, for example, can hear a higher pitched whistle. So amplitude is the height of the wave, so how big that is, and we perceive that as how loud it is. And that happens when more air is pressed more forcibly against your ear. Because what the sound is pressure waves that are moving through the air. So I have a little speaker. Well, right now I've got a little speaker literally in my ear. And so there are pressure waves moving the little bit of air that's in my ear in between and then moving the eardrum, moving some bones, and then moving fluid around that's moving hair cells. And then those hair cells activate and send a message to my brain that I'm hearing a noise. Timbre is that most sounds have multiple wavelengths. So this is kind of like the purity of light kind of thing. So this is the tone saturation. So is it a pure, clear, crisp sound, or is there multiple kinds of sounds together at one time? So here's the structure of the ear. So we've got a speaker outside the ear. There's some air here in the ear canal. So that pressure wave is pushing that air, and it pushes that air up against the eardrum. Probably you all know at least to that point in the structure of the ear. And then connected to that eardrum is a couple bones. So this is actually a key features of mammals, that we have these specific kind of ear bones. So it's not, I learned at the Museum of Natural History that it's not just hair, mammals having hair, it's also our specific ear structure of the bones. So not really relevant, but kind of tangentially related. So the hammer, anvil, and stirrup all then push this, so that's all making up the middle ear, and push this little essentially window to push fluid through this area, the cochlea, and then in there are the hair cells that will then move depending on where on the cochlea is that pressure is being moved. So we'll watch some videos too about this. So let's do, I think this one is the best for what we want to look at.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration, and sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. Two structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a complete...
So one thing we probably won't have time to cover today is that senses like where your body is, kinesthetic senses, your sense of balance. A lot of that is due to hair cells in this part of the inner ear, so within this bony labyrinth. So the auditory signals we get are coming through this side of it, so this kind of spirally snail looking side, the cochlea. But our senses of balance, of where our body is, if we're leaning over, those kinds of things, a lot of that is from hair cells on this side of the ear. So pretty amazing that this little structure does so many different things with these mechanoreceptors. I think it's really interesting that the same kind of receptors that give you the sense of touch, like pressure, it's very similar cells that give you your sense of hearing as well. And we'll see why now.
Closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us. scala vestibuli.
So I know it's a good song. I stopped it too early, but I just wanted to point out here. So here at the top, well, from at this angle, the more tightly coiled spiral part, which at this image is at the top of our image, that is where, if that is vibrating, we get the sense of a low pitched frequency, a low pitched sound. If this part near the bottom of the image that we're looking at, if this is vibrating, we get a sense of a high pitched frequency. So it's pretty amazing to me that inside this small structure in our little inner ear is an even smaller organ with these tiny little hair cells, that because this specific part of it is vibrating, we hear a high-pitched noise. So because of the pressure of the hair cells against that other membrane, and that is how we evolved our sense of sound. That's how we sense sound. And then from that, we perceive it. We perceive music, we perceive everything, starting from that sense, just from that vibration, moving specific hair cells. So hair cells up here moving, you got a low sound, hair cells down here moving, you've got a really high sound. It's the sense of the visual sense for me makes a lot of more intuitive sense, because knowing how a camera works, things like that. The auditory sense still kind of boggles my mind sometimes, that we evolved a sense of sound based on pressure of vibrations in the air. It's pretty amazing. So here's another shot. So here, this is where low sounds are coming from, high pitched sounds, another cross section of the hair cells. So again, that is a specific receptor cell that when it gets the pressure, this part of the cochlea is vibrating, it then activates and sends that signal to the brain. So pretty neat. Cochlear implants are specifically there to replace damaged hair cells. So I wish I had made this picture bigger because it makes it a lot more clear. So essentially you have a microphone and a transmitter, and then you have electronics stimulating any working auditory nerve cells, so any hair cells within the cochlea. So there are literally little electrodes within the cochlea, and a cochlear implant is activating hair cells that are working. So it doesn't amplify sound, it just makes any working hair cells work more efficiently or work better. So that's what a cochlear implant is, and that's very different than a hearing aid, which is just trying to amplify the sound, make it bigger. Yeah. So the place theory of pitch perception is basically just depending on where the vibration is happening within that cochlea, within that spinny structure, snail-looking structure, that will give you the frequency. So with that, our neurology, we talked about the brain, where different aspects of the brain are affecting things. A lot of our senses of touch, from our hands, our face, genitals, feet, it's all going essentially to a part in your brain like about right here. So as you can see, this is the kind of the middle of your brain. This is to the side. So right here, kind of near the top middle, is where kind of I get my sense of feeling of where my core is. About right here is where my sense of touch from my head. So I'm literally touching like on my head where my neurons are getting the message from my head. Maybe this doesn't actually make much sense because it's a very little image. But basically, as you can see, there's a lot more neurons, a lot more space for the eyes, the nose, the face, the lips, than say other parts of our body for feeling the sense of pressure. So that's why things like hands, we have a lot more dexterity, a lot more feeling in them, because there's so much space dedicated to that in neurons, so many more neurons dedicated to doing that. Yeah. So thermoreceptors are specific neurons that detect temperature under our skin. We have receptors specific to warm and receptors specific to cold. Pain actually comes through the same cells as touch and temperature. So it's essentially those same cells if it gets a really intense sense, like if it's really sharp, hard, intense, it will then send a trigger of pain rather than just cold or rather than just pressure. But it's through the same exact receptors. There's not a specific pain receptor. It's these same thermoreceptors or these Meissner's corpuscles. I probably said that wrong. So with that, to send that pain signal, it's a very high absolute threshold comparatively. So you could poke your wrists, for example, and you're activating your touch receptors, those mechanoreceptors, or you could really vigorously tap your wrist, and then you might get a pain sense. It takes a lot higher absolute threshold for those nerve cells, for those receptor cells, to send that pain signal. Taste is taste buds. You've probably heard that term. We have sensitivity to the tastes of salt, sour, sweet, and umami. Umami is definitely my favorite one. If you've ever heard of MSG, monosodium glutamate, that's umami. It's in things like fish, cheese, other meats, and it's definitely my favorite one. Smell essentially evolved to help keep us safe from rotten food. So it helps us detect different things in the environment. In a lot of mammals, it helps with mate selection in sensing and smelling hormones and finding mates. It's not clear if that's the case for humans. There's evidence that might suggest that humans can detect hormones from other humans or specific pheromones. There's other evidence that it's not really that doesn't happen. So that could be another question paper if it's something you're interested in. You might find one paper research article that says, yes, humans can detect pheromones, or it seems like they can. You might find another saying, nope, it doesn't seem like they can. So yeah, I think it's interesting. It's definitely not as important for humans as it is for other mammals, though, the pheromones. So I've got a hand. Lindsay, go for it.
Just curious if you know the answer to this. What about the coronavirus causes you to lose your sense of taste and smell? Do you know?
That is a good question. Off the top of my head, no, I don't remember. I know I've read it, like why it is, but I don't remember off the top of my head. I don't think it actually like damages the receptor cells. I think it's a way that we're perceiving it because of the virus. But if anyone else knows the answer, feel free to chime in. But I can definitely look that up. I'm actually going to write it down. Yeah, that's a good relevant question with sensation and perception that I can look up. Yeah, sorry, I don't know that. I do remember reading it when the whole pandemic first started, but yeah, it's left my memory now. But really good question. If I remember right, though, I don't think it actually damages receptor cells, but I think it has to do more with how we are perceiving the signal, the sensation of those tastes. But yeah, I'll look that up for next time, or maybe just send you an email about that. So kinesthetic sense is one of the last that we'll mention. This is how you know where your body is in space. So if you are a dancer, you probably have a lot better kinesthetic sense than someone who's not a dancer. It lets you know where that your arm is bent or not bent, that's behind you, things like that. And that is coming through receptor cells in muscles and tendons. So with more practice, those receptor cells get more use. Your brain knows better where those things are. That's how you might know in a dark room that your hand is near the light switch, even if you can't actually see it. Vestibular sense, this is one I mentioned earlier when we were talking about auditory signals. So these semicircular canals have to do a lot with balance and acceleration, knowing that you're tipping over, knowing that you're moving left, right, forward. If you're on a roller coaster, you're getting those semicircular canals moving. This is one reason some people feel motion sick with like VR, because they might be watching visually, they're getting the sensation that they are moving from the visual, like moving around them, but these vestibular senses are not giving them that same sensation. So it can actually make some people feel sick because of that. Because it's kind of confusing your brain. You're getting one sense that's giving you the sense that you should be moving, but your vestibular organs are saying, no, I'm not actually moving. And so that can be a little confusing. And that is the last for sensation and perception specifically. Are there other questions, though, about things that we've talked about so far today? With the extra time after class, I'll definitely look up again why coronavirus changes smell and taste, see if that's sensation, perception, or some combination of both of those, because that's a really good question. But are there other questions that any students have, or now that we're at the end of class and we're finished with chapter 3 and the exams coming on Friday and Saturday, any questions about that or how that will work logistically? So as a reminder, your exam does open on Friday. Basically, as soon as Friday starts, the exam is open and available, and it closes just before midnight on Saturday, the 19th. So make sure you take it in that time. It's a 70-minute time limit. Oh, looks like we did get a question in the chat. Yep, if you don't see your score right after you're done taking the quiz, email me right away, because that means that there's something wrong with the settings. But you should see your score right away, because it's all entirely multiple choice. So that'll be your grade on the exam, unless I've made a mistake on the exam. It's possible that maybe I've miskeyed a question. So definitely go through the questions then, see what it's marked correct and wrong. Because if there's a mistake that I've made, if it's marking something wrong that shouldn't be, then your grade could go up because of that. Because then I can fix it. All right, well, good luck on that exam, students. I know you'll do really great. I know it's pretty close to the time where we're talking about chapter 3. So definitely make sure you review maybe this lecture. I will post it up tomorrow. It takes a little while to upload it to YouTube, so it'll probably be up on Canvas tomorrow. So review this one, review any of the previous lecture videos as well. Go over your notes, go use that study guide. Get in groups to go over the study guide. You can meet here in teams at any time, essentially. You can email some students and be like, hey, do you want to meet up on Thursday at 3 in the afternoon and we can go over the study guide? You could definitely do that together. So thanks for being here. Thanks for your questions. I'm glad I got stumped by a question too, or 2 questions. So good work and good luck on the exam.